Good morning, students. We will continue the topic of chemistry of lipids, competition number 4.1, and today's topic is biological importance and functions of major phospholipids and sphingolipids. As we have studied in the chemistry, phospholipids are mainly made up of alcohol, glycerol, or sometimes the alcohol may be sphingosine, with fatty acid, phospholipids, and nitrogen base. So this is the structure of phospholipid, where alcohol is glycerol, fatty acids phosphate group, and nitrogen base is choline. These phospholipids are amphipathic in nature. That means they have hydrophobic part as well as hydrophilic part. Here the hydrophilic part or head is formed by glycerol, phosphoric acid, and choline, whereas the hydrophobic part or tail is formed by the fatty acid. So this is the diagrammatic representation of phospholipid molecule on the left side, where the head is formed by choline, phosphate, and glycerol molecule, and the tails is formed by fatty acid molecules. Now, due to this amphipathic nature of phospholipids, in the aqueous medium, these phospholipids get arranged themselves in such a way that their hydrophobic tail will hide inside, and their hydrophilic head faces the aqueous medium, forming a lipid bilayer on which on either side there is aqueous medium, that is inside the cell and outside the cell. So the hydrophilic portion of the phospholipid faces the aqueous medium. So when this structure of cell membrane is formed, it allows the hydrophobic substances like carbon dioxide, oxygen to diffuse through it easily, but it does not allow the hydrophilic substances to passes through it. Hence the cell membrane is called as semi-permeable membrane. So phospholipids plays an important role in the formation of cell membrane and its semi-permeable nature. Next importance of phospholipid is these phospholipids forms micelles. Again, in the gastrointestinal tract, after digestion of the fat, the phospholipids which are present in the GIT or aqueous medium of GIT, they get arranged themselves in such a way that they form a circular structure in which their hydrophobic tails hide inside and their hydrophilic head present outside, facing the aqueous medium of the GIT. In this process, they trap a small space at the core in which the pure hydrophobic lipids like triglycerides and cholesterol esters are caught. When this structure or when these micelles are formed in the GIT, then these micelles easily enters into the epithelial cells of GIT. Thus, phospholipid plays an important role in the absorption of triglycerides and cholesterol esters, because when these micelles enter into the epithelial cells, triglycerides and cholesterol ester also enter along with them. So this is formation of micelles which helps in the absorption of lipids. Now the next importance of phospholipid is they also plays an important role in the formation of lipoproteins like chylomicrons, VLDL, very low density lipoprotein, HDL, high density lipoprotein, and LDL, low density lipoprotein. If you see the structure of these lipoproteins have internal core where pure hydrophobic lipids like triacylglycerol and cholesterol esters are present, surrounded by a amphipathic lipid like phospholipids and free cholesterol, and whole structure is then wrapped by the proteins. So it is a combination of lipids and proteins. So phospholipids forms the peripheral part of this lipoprotein because of their amphipathic nature. Again, they hide their tail inside and their hydrophobic head remains outside, plays an important role in the formation of various lipoproteins. Phospholipids also plays an important role in the formation of liposomes. These liposomes are artificially prepared by using phospholipids. They are double membrane structure, circular, with their hydrophobic part is hiding inside and hydrophilic part is facing outside and into the lumen. Here also liposomes trap a space in the core where you can deliver the hydrophilic drugs, proteins, enzymes and genes. After preparing these liposomes are given to the persons and they are specifically absorbed by the various tissue. That means various liposomes are prepared for various tissues so that these liposomes enters or taken up by the specific tissue only, and in this way you can deliver the various drugs, proteins, enzymes and genes to that organ. So this type of liposomal applications are seen in cancer therapy, antimicrobial therapy, gene therapy, delivering vaccine and some diagnostic imaging techniques. Now we will study individual phospholipid. In short, first phospholipid is phosphatidyl choline. All of us know that. The importance of this phosphatidyl choline is it is the important constituent of surfactants in the lung. Normally, the lung function depends upon the surfactant. These surfactants are produced by the epithelial cell. Inside the alveoli, these surfactants forms a thin layer so that when the air comes out of that alveoli, this alveoli does not collapse. That means they decreases the surface tension from the inside of the alveoli and prevent collapse of lung alveoli. Now this surfactant contains dipalmitoyl lecithin, phosphatidyl glycerol, cholesterol and protein A, B and C, where dipalmitoyl lecithin is the main composition of surfactant. When the surfactant is not synthesized in sufficient quantity, then the lung alveoli may get collapse, leading to a disease called as respiratory distress syndrome, which is the important cause of neonatal morbidity. Here you should know that the surfactant phosphatidyl choline is synthesized by the lung tissue after 28 weeks of gestation. Before 28 weeks, the lung will synthesize Springomyelin, which does not have any effect in lowering the surface tension of the lungs. But after 28 weeks of gestation, slowly the synthesis of Springomyelin decreases and synthesis of dipalmitoyl lecithin increases, which indicates the maturity of the lung tissues. Here the ratio, LS ratio, that is lecithin to Springomyelin ratio in the amniotic fluid is an index of fetal lung maturity. If the ratio is 2 or more than 2, then it indicates lung maturity. If the ratio is less than 2, it may lead to respiratory distress syndrome. So this is the respiratory distress syndrome. Just now I have explained. It is generally seen in premature infants where the synthesis of dipalmitoyl lecithin is less, and it is the cause of neonatal morbidity. Next phospholipid is phosphatidyl ethanolamine or cephalin. It is found in biomembranes and plays an important role in it. Next is Springomyelin. Here the alcohol is not glycerol, instead of which the alcohol is sphingosine and the nitrogen base is choline. These Springomyelins are found in large quantity in nervous tissue or nervous system. Depending upon the attachment of fatty acids in the sphingomyelin. Different sphingomyelins are found in the body. The common fatty acids which are found in the sphingomyelin are lignoceric acid, nervonic acid and cervonic acid. So this sphingomyelin, just now we have studied the ratio of lecithin to sphingomyelin in the amniotic fluid, gives us the idea about the fetal lung maturity. This LS ratio is also important in bile. Next phospholipid is phosphatidyl inositol bisphosphate. It is found in biomembranes. It also acts as a secondary messenger for water soluble hormones which cannot enter into the cell. Plasmalogens are found in biomembranes, brain and muscles. Phosphatidyl glycerol is also called as cardiolipin or diphosphatidyl glycerol, where two glycerol molecules are joined together by phosphoric acid. It is the major phospholipid found in the mitochondrial membrane. It is also extracted from myocardium, hence it is named as cardiolipin. Decreased cardiolipin level may lead to mitochondrial dysfunction, heart failure, hypothyroidism, and in some myopathies also you can find the decreased phosphatidyl glycerol level. So this completes the biological role and importance of phospholipids in the body. Thank you very much.